Dear students, here Dr. Sivagama Sundari, Assistant Professor, Department of Life Sciences, Krish Jayanti College. So, this video lecture tells you about the structure of seed. So, we will move on to the general terms. So, embryogenesis or embryology. Seed is related to embryo, which is having that embryo which is developing inside the seed. So, embryogenesis or embryogeny is the study of subsequent period of development in which the zygote undergoes a complex series of cellular as well as morphological changes. So, after these changes, the embryo is converted into a diploid multicellular eukaryotic organism or a whole plant. So, we call that process as embryogenesis or embryogeny. So, embryology is a science that deals with the study of embryo from the time of fertilization. So, this is how the stage of embryo development happen. This is an example for dicot, monocot also it will be same, but you cannot able to see the complete structure of like octant, globular, triangular, heart and torpedo shape, because compared to monocot, dicot it has two cotyledon. So, first fertilized egg, after that it will develop into two cells, apical and basal cells, then it move on to octant stage, globular stage, triangular stage, heart stage, torpedo shape, then mature embryo which has the cotyledon. So, this stages in cellular as well as the morphogenesis we call it as embryogenesis. A person who perceive the study of embryo in a angiospermic plant, we call it as embryology. The study deals with the embryo, we call it as embryology. So, structure of seed. Seeds of different plants may vary in many ways, but the basic anatomy of the seed remains the same. A typical seed consists of the following parts which you already aware: micropylar region, radicle where the root emerges out, epicotyl where the shoot emerges out, cotyledon, whereas in dicot you have two cotyledon, monocot it will be only one, we call it as scutellum, and testa nothing but the covering of the seed. Moving on to the elaborate structure. Testa, it is the outer coat of the seed that protects the embryonic plant, which means the developing embryo which is present in the seed, it will be protected by the testa. That is outer coat. Tegmen, it is the second coat of the seed, that is the inner line of covering, we call it as tegmen. Then micropylar region which is present in the ovule after fertilization. Because of the post fertilization changes, it is a tiny pore in the testa that lies on the opposite of the tip of the radicle, permits water to enter the embryo before active germination. So, that is what when you want to germinate any seed, you have to spray some water. So, that water will enter into the micropylar region, that will dilute the complex substance into simpler one, that enables the seeds to germinate. Hilum, scare left by the stalk which attach the ovule to the ovary wall before it become a seed. Plumule, this is the embryonic shoot, it appears as a bud which will give rise to shoot and the remaining structures in the plant. So, continuation, coming to the cotyledon. In some plants, this contain high quantities of starch and will provide a source of food for the developing embryo prior to germination. That is why it is recommended the germinated seeds are good for your protein source. In other plants, this role is performed by an endosperm, it is a nutritive tissue. In monocotyledon, there is just one cotyledon, where in dicots there are two. Coming to the radicle, this is an embryonic root which will develop into the primary root of the plant. It is usually the first part of the embryo to push its way out of the seed during germination in order to derive the nutrition. Then endosperm, it is a nutritive tissue. You might have heard about the double fertilization. So, the second fertilization of the nuclei, male nuclei, will happen in the polar nuclei that develops into a triploid structure, endosperm. We call it as nutritive tissue that gives the nutrition to the developing embryo. So, these are the parts comprising a monocot and dicot seed. Only difference is the number of cotyledons in monocot and dicot. So, moving on to the functions of the seed. Seeds help in the germination of the new plant. That is the only source of genetic stock. After the fertilization of male and female germ unit you are getting. So, that is the only genetic stock to you. You can start the next generation. The seeds contain full of food reservoirs in the form of cotyledon and endosperm. It is full of starch. Then the seed coat is protective, testa and tegmen, which protects the embryo inside. So, this video I am sure that make you aware about the embryogenesis, embryology, how the structure of seed look like, what are the compartments that are present in monocot and dicot seed, and functions of seed as well. Thank you.